First of all, I would like to thank you for inviting me to the seminar. It's my great honor for me to present my research area. I am Hongjo Lee, Assistant Professor, Division of Animal Science, University of Missouri. Today, I'm going to present my research, which is entitled Avian Genetic Engineering, Expanding CRISPR Toolbox. I would like to start my presentation with the reason why I'm studying avian genetic engineering. In the industrial field, the avian species has been used as an important protein resources coming from their meat and eggs like other livestock. And the avian species have been recognized as an important animal model in diverse research fields, including developmental biology and immunology, due to their unique developmental patterns. With a bunch of studies and invention of technology, the value of avian species has been increased not only for the poultry industry, but also in academia. Nevertheless, there are still limitations to be overcome, such as disease spreading and animal welfare, which prevent their efficient usage. So I thought genetic engineering technology can be one of the alternatives to solve those issues. To produce genome-edited cows, pigs, sheep, and goats, as far as I know, we need to inject DNA such as transposon, viral vector, and CRISPR-Cas9 to zygote, or transplant in vitro modified cell line into zygote or enucleate oocyte. However, those techniques are hard to apply in avian species. Please look at the reproductive tract of chicken and its embryo development. A chicken egg is ovulated with a large yellow yolk, and just right after ovulation, an oocyte fertilized at the infundibulum. The embryo develops and differentiates in the oviducts for 25 hours before oviposition. Basically, we need a one-cell stage zygote or stem cell population for direct DNA injection or nuclear transfer. However, as you can see, it hurts to get a one-cell stage embryo or oocyte from this long reproductive tract. Even if you extract zygote by abdominal massage, the large yellow yolk limiting embryo manipulation. As an alternative, early studies on avian genetic engineering use blastodermal cell for germline transmission. However, the efficiency was extremely low because those cells are already differentiated in the oviduct, although the cell contains a few germ cells. Furthermore, there are no well-established stem cell line in avian species. We cannot adopt the stem cell transplantation method either. To overcome those limitations, researchers found an alternative germline transmission system for avian species using primordial germ cell, PGCs. PGC is a precursor cell of sperm or egg, and it contains whole genome information because it is one of the germ cell type. Notably, bird PGC have unique migration activity. The PGC localized at the center of blastodermal cell move to semicircular shaped extra-embryonic region called the germinal crescent area, then migrate to the embryonic gonad through a blood vessel, unlike mammalian PGC. Using the characteristics, the early researcher developed a PGC-mediated germline transmission system. First of all, we can isolate PGC from embryonic gonad To isolate PGC, the embryonic gonad attached to mesonephros were extracted by a sharp tweezer. After dissociation of the gonadal cell was then cultured in PGC media. Once the number of PGC reaches enough number, we can transfect genome modification vectors for establishing genome-modified PGC line. Then the PGC were transplanted into the recipient embryos through an embryonic blood vessel, like this video. The video shows how to extract blood from embryonic blood vessel, but we can put PGC into an embryonic blood vessel the opposite way. After the PGC transplantation, the recipients hatch and reach sexual maturity. Then the recipients are validated whether they can produce donor PGC-derived progenies by testicles. Using the method, generating the first genetically modifying chickens was reported in 2006. They cultured PGCs and modified their genome and generated donor PGC-derived progeny. The germline transmission efficiency was greatly higher than other germline transmission systems. In our lab, we also established in vitro PGC culture method. In our lab culture condition, PGC proliferated well with a germ cell specific marker expression. And the culture PGC derived progeny were successfully generated by PGC transplantation. Then we started to adapt diverse genetic engineering technology into the PGC-mediated germline transmission system. First of all, we applied the piggyback transposition to the PGC-mediated germline transmission system. Piggyback transposition system is one of transgenic biotechnology that allow us to insert gene of interest flanked between terminal lipid into genomic DNA. By transfection of piggyback transposon containing GFP gene with a piggyback transposase, we successfully established GFP expressing PGC line, and subsequent PGC transplantation allowed us to generate transgenic chickens which express GFP in their bodies. Next, we applied TALEN, one of the programmable genome editing technologies, to PGC-mediated germline transmission system. In this paper, we targeted one of the major egg white protein, ovalbumin, and succeed to generate ovalbumin gene mutated chickens. By sequencing analysis, we could find that indel mutation were induced by TALEN on the targeted ovalbumin gene in the donor PGC-derived chickens. Then the invention of CRISPR-Cas9 has changed everything. CRISPR-Cas9 is third generation of programmable genome editing technology derived from bacterial immunity. The technology allows us to induce double-strand break on the target region by simply introducing Cas9 protein and guide RNA complementary to targeted nucleotide sequences. Due to its convenience and versatility compared to previous programmable genome editing tools, single finger nuclease or TALEN, it has been widely applied in diverse living organisms for diverse purposes, including disease control and improving traits. CRISPR-Cas9 inventors Dr. Charpentier and Dr. Doudna were awarded the Nobel Prize in Chemistry 2020 And it is not surprising that it takes only 8 years to be awarded the Nobel Prize if you can imagine the huge impact of CRISPR technology on our world. Now, the CRISPR technology has been expanded by combining other effector proteins as well. Now we can change genetic information using Cas9 and change epigenetic profile using DNA methyltransferase and histone modifiers, and also regulate gene expression by modulating DNA accessibility, which were called CRISPR activation, CRISPR-A, CRISPR interference, CRISPR-I. In my research, I would like to apply this revolutionary CRISPR technology to avian species for diverse purposes, including enhancing animal welfare and production trait, and acquiring disease resistance, and animal model development for sex determination and germ cell tracing. The first application of avian genetic engineering in my research was Z chromosome targeting for animal welfare. In the poultry industry, especially in egg production, they only need female chickens which can lay eggs. So the male chick are killed after hatching. The male chick culling has been a problem in terms of animal welfare as well as economic losses. So we thought if we can produce transgenic chicken expressing GFP from the G chromosome, we can identify chicken sex before hatching with a GFP detector. Male chicken has GZ chromosome and female chicken has G double chromosome. So if we insert GFP gene into the G chromosome, and the transgenic female chicken only can produce GFP expressing male chickens and wild-type female chickens by mating with wild-type male chickens. So I thought the strategy could be one of the alternative chicken sexing model. To conduct targeted gene insertion into the Z chromosome, we applied CRISPR-mediated non-homologous end joining NHEJ method, which is previously adapted in zebrafish, to our chicken PGC-mediated germline transmission system. By transfection of CRISPR-Cas9 vector with a donor plasmid containing guide RNA targeting sites, the Cas9 protein can linearize the donor vector and induce double-strand break on the targeted region of the chicken PGC genome. Then, the linearized donor plasmid can be inserted into the targeted region in which double-strand break occurred. The genome-modified chicken PGC were cultured, selected, and transplanted into lesbian embryos as previously described for generating transgenic chickens. To identify whether the CRISPR-NHEJ method is working in avian cells or not, we first apply the strategy to the chicken somatic cell line, DF1. We targeted the intergenic region of Z chromosome, preventing endogenous gene expression change. It was expected that the linearized donor plasmid can be integrated into the targeted region of the chicken G chromosome by co-transfection of CRISPR-Cas9 vector and the donor plasmid, and the targeted cells should express GFP from G chromosome. We transfected CRISPR-Cas9 vector and donor plasmid containing GFP gene and G guide RNA targeting sequences. After transfection and drug selection, we successfully established a GFP expressing cell line, and targeted gene insertion was confirmed by genomic DNA analysis. And we found that the strategy can be applied to chicken PGCs as well, and finally succeed to generate G chromosome knock-in chicken by transplantation of the G chromosome targeted PGCs. From this study, we could find that CRISPR-NAGEJ method is a versatile tool for targeted gene insertion in chicken. As a second application of avian genetic engineering in my research was inducing double muscling and modification of egg protein composition. In this research, we used base editing technology was developed by two independent research groups. Base editing is one of the expanded CRISPR toolbox kits, which combines cytidine deaminase with a dead Cas9 or nickase Cas9 for nucleotide substitution. Using the base editing technology, we would like to induce nucleotide substitution C to T, causing a generation of premature stop codons on the myostatin gene, which is muscle growth inhibitor. By transfection of base editing vector targeting myostatin gene, we successfully induced nucleotide substitution on the targeted region, generating premature stop codon. The strategy was also applied to one of the major egg white protein overtransferrin to modify egg white protein composition. As same as myostatin gene base editing, we found that the overtransferrin gene was also successfully modified by base editing vector transfection. Then we transplanted the base edited PGC and succeeded in producing myostatin or overtransferrin gene modified chicken. In addition, in this research, we also found the base editing technology can induce unwanted nucleotide substitution or indel mutation as well as desired nucleotide substitution. Indeed, as you can see here, we found that there was significant difference in base editing activity between DF1 and PGC at both genes. To explore the phenomenon, we validated repair pathway related gene expression and found that the base excision repair pathway is significantly elevated in PGC compared to DF1. So we modulate the UNG gene expression, which is the most upstream gene of the base excision repair pathway, by overexpression and knockout. Then identify that overexpression of UNG gene reduced the base editing activity, and knockout of the UNG gene increases editing activity. In this research, we found that we could generate myostatin gene or overtransferrin gene modified chickens by one of the CRISPR toolbox base editing, but at the same time, we could know we can promote the base editing activity by modulating the base excision repair pathway. For the next stage study, I also applied CRISPR toolbox in chicken system for acquiring disease resistance. Specifically, we would like to acquire the disease resistance against to avian leukocyst virus, ALV. ALV is a retrovirus and is divided into 6 subgroups. The virus transmits horizontally and vertically, and the virus infects and can lead to cancer in chicken. The virus has been eradicated in the Western world. However, the virus still causes economic losses in China, which has a big poultry market. As same as other viruses, the ALV has specific host receptor for each subgroup. ALV subgroup A specifically bind to the TVA receptor, and TVV is specific for ARV subgroup B, D, and E. ARV subgroup C specifically binds to TVC receptor, and ARV subgroup J uses chicken NHE1 receptor for viral entry. Therefore, we hypothesized that if we disrupted the receptor gene of chicken, the chicken could get resistant to the virus, like a CD163 knockout pigs for perceived prevention. To prove the hypothesis, we constructed CRISPR-Cas9 plasmid targeting the TBB gene, encoding TBB receptor, which is the ARV subgroup B host receptor. By transfection of the CRISPR-Cas9 vector, it successfully induced in the mutation on the targeted region. Then we infected the GFP-expressing virus to the DF1 clones. As a result, we found that the clones having mutations on the TBV gene showed significant reduction of viral infection. From this research, we found that CRISPR-Cas9 mediated viral host receptor gene knockout confers resistance against two virus in chicken. For the next step, we disrupted 3 different viral host receptor genes sequentially and found that the clones that have mutations on multiple viral host receptor genes confer resistance to multiple ARV viruses. In this research, we also found that the 3 different ARVs do not share their own receptors. Using the method, we generate the genome-edited chickens, and now the chickens are validating whether they are resistant or susceptible to ARV viruses. And we apply genetic engineering technology in the developmental biology area as well. Indeed, I have great interest in avian developmental biology, especially in sex determination and germ cell development. As I mentioned in Z chromosome targeting research, chicken has 2 sex chromosomes, Z and W. Male has ZZ and female has ZW, and the distinct morphological difference between the sexes is gonad development. As embryogenesis proceeded in female, only the left gonad is growing and become functional, but the right gonad started to regenerate and finally it will be disappeared. On the other hand, both left and right gonad grow symmetrically and become functional in male chicken. This sexually dimorphic phenomenon is known to be caused by the chicken sex chromosome, and the DMRT1 gene located on the G chromosome is the most promising candidate for asymmetric gonad development in female chickens. Because early research about DMRT1 gene showed that downregulation of DMRT1 gene induced asymmetric gonad development in genetically male chicken. However, due to the lacking technology, they could not further validate the gene function at the later stage of embryogenesis and after the hatching stage either. To identify the DMRT1 gene function, we also apply CRISPR NHEJ method to chicken cells for disrupting the DMRT1 gene. To disrupt DMRT1 gene expression, we conducted targeted gene insertion into the translation start site ATG of DMRT1 gene. By introducing the CRISPR-Cas9 vector targeting DMRT1 gene and donor plasmid containing GFP gene into the cell, we successfully established the GFP-expressing DF1 and PGCs, and we also confirmed that the donor plasmid was integrated into the targeted region of the cell by sequencing analysis. The PGC were transplanted into recipient embryos, and DMRT1 gene-disrupted chickens were produced. As expected, the transgenic male chicken have ovary-like testes, like a wild-type female, and we could see clear cortex and medulla structure by histological analysis. In addition, we could detect a reduced expression of DMRT1 protein with increased expression of female granulosa cell marker FOXL2 in the DMRT1 disrupted male chicken gonads. However, unexpectedly, the adult male transgenic chicken failed to develop functional ovaries and oviduct and showed impaired hormone synthesis and germ cell losses. These results suggest that DMRT1 gene disruption alone could not induce complete gonad feminization in chicken. In this research, we further analyzed molecular mechanism for gonad feminization. By comparing transcriptome profile between wild type and DMRT1 disrupted chicken's gonad, we found again the male transgenic chicken gonads are not fully feminized transcriptionally. Moreover, we found several novel genes relating to DMRT1 dependent gonad feminization pathway. We also found that there are many novel genes, including RNC RNAs, showing ovary-specific expression may be involved in gonad feminization pathway in DMRT1 independent manner. For the next step, I would like to make a germ cell tracing model. As an avian genetic engineer, the germ cell is one of the most important cells in chickens. We are always thinking about germ cell. Those growing PGC earlier PGC or the genetic or epigenetic events are conserved in avian species. Can we make a sperm or oocyte from these cultured PGC? However, whenever we ask ourselves those kind of questions, we face the reality. We don't have information enough about germ cells because there are no surface antibodies of germ cell that allow us to isolate them. We only can isolate the early stage. We only can isolate germ cell at the early stage of embryogenesis when just right before PGC differentiation. To address these challenges, we decided to produce a germ cell tracing model that allow us to isolate and analyze germ cell during whole embryonic developmental stages. To develop germ cell tracing model, we tried CRISPR-Cas9 mediated gene tagging. We thought if reporter genes such as GFP were ligated into germ cell specific marker genes, we could isolate the GFP expressing germ cell at every developmental stage using the reporter gene expression without altering the germ cell marker gene expression. As a germ cell marker gene, we selected DAZL gene, which is germ cell marker in both sexes from early stage of embryogenesis. To conduct gene tagging on the gene, we constructed the CRISPR-Cas9 vector targeting the last intron of the DASO genes in the donor plasmid containing part of last intron and exon of DASO gene linked to GFP coding sequences by 2A self-cleavage peptide. By transfection of CRISPR-Cas9 vector in the donor plasmid, we could detect GFP-expressing PGC. After drug selection, we successfully established the genome-modified PGC line and succeed to generate other GFP transgenic chicken by transplantation of the GFP-expressing PGCs. We further validated transgenic chickens and found that GFP expression at germ cell from germinal crescent and the embryonic gonads at early stage and later stage of embryos were detected in the transgenic male and female chickens. Then we collected germ cell at different time points, isolate GFP-expressing germ cell by FACS, and analyzed transcriptional profiles by single-cell RNA sequencing. By single-cell RNA sequencing, we identified 4 and 5 transcriptionally distinct germ cell stages at male and female germ cell, respectively, during embryogenesis. And we also identified male germ cell have clear one trajectory developing to spermatogonia, but female germ cell have 2 distinct trajectories, one for apoptosis, one for meiosis. In addition, we further compared chicken germ cell with a human germ cell, and we could find conserved or differential transcription profile between 2 species. Using the expanded CRISPR technology, now I'm trying to expand my research at the University of Missouri. Since agriculture in Missouri has huge effects on state economy, so I have planned to apply genetic engineering technology to the industrial field. It could be animal welfare, and actually, honestly, I have a plan to make a W chromosome targeting transgenic chickens. Then we can identify chicken sex before hatching. Also, we could enhance production trait, including muscle development and growth rate or feed efficiency. And also, we could modify egg protein composition by modifying egg white or egg yellow protein genes. Using CRISPR toolbox, I also would like to control disease such as the influenza virus. Most recently, there are outbreaks of influenza virus in the United States. If we apply genetic engineering technology to acquire disease resistance, especially in avian influenza virus, it could contribute to industrial field by preventing disease transmission. Indeed, I have experience with an influenza virus project. We applied CRISPR technology to disrupt influenza host factor AMP32A gene, which help influenza virus proliferation. By simply knocking out AMP32A gene by CRISPR-Cas9 like ARV virus receptor gene, we established the disease-resistant cell line. Moreover, in this project, we modified the surface glycan structure by overexpression of V4 guarantee 2 for altering virus susceptibility. As you can see here, the modified cells showing lower virus susceptibility compared to wild-type cell line, which means confer resistance again to avian influenza virus. And I said previously, I have great interest in germ cell biology. As mentioned before, the germ cell research in birds has been largely limited due to the lack of surface markers, but now we have germ cell tracing model, Tazo GFP chickens, and single-cell RNA-seq database from the germ cell at diverse developmental stages. And moreover, we secured chicken PGC chip-seq database about histone profile. Based on these chickens and database, I think we can explore genetic or epigenetic events occurring in germ cell development during embryogenesis. Moreover, it could enhance the understanding of germ cell. Maybe we unable to develop induced germ cell in vitro. One another project what I'm interested in is developing novel gene regulation platform. Not only inducing double-strand break, CRISPR-I and CRISPR-A can regulate gene expression by binding to promoter, enhancer, or repressor regions without double-strand breaks. Therefore, if we establish those second line, we can use them for diverse researches by simply adding guide RNA targeting a different set of genes to diverse cell types of chickens. I'm a new faculty member of University of Missouri. So most of research has been done at Seoul National University under Professor Han's lab, under Professor Han's supervision. Therefore, I would like to acknowledge my previous labs, and also I would like to acknowledge my postdoc lab at Penn State University. And also I would like to acknowledge my collaborators for single-cell RNA sequencing and biometric analysis, Dr. Min-Seok Seo and Dr. Jong-Kyung Kim. And also, I would like to acknowledgement of my funding agency for grant funding. Last but least, now I am establishing my program at University of Missouri. The University of Missouri is specialized state university, and there are huge great support for researchers. Particularly our MU Livestock Engineering team, MU team, is the leading research group for livestock gene editing in the United States of America. So it is perfect place to conduct research relating to gene editing in livestock. Therefore, please let me know if you are any interest in the genome editing research, not only for avian species, but also other livestock such as pigs, cows, sheep, or the other livestock animals. Recently, we have received 8 million grant for livestock center, which can raise a lot of livestock for translational research for human xenotransplantation. So there are also so many opportunities to be receive a lot of amounts of grants in University of Missouri. So again, please let me know if you are interested in genome editing technology. Here is my email address, and I'm looking for postdoc, researchers, and grad students. Thank you for listening.